So we're going to move on to our next speaker, and Dr. Lewis, would you like to run the introductions?
Yes. So it's with great pleasure that I introduce to you our next graduate student speaker, Mark McDonald. Mark comes to us from Crossville, Tennessee, which is about 70 miles west of Knoxville. He completed his undergraduate there, getting a bachelor's of science in biology with a microbiology focus. He completed his master's degree with me at Texas Tech University. And then after completion of that, he started at A&M with both Dr. Terry Gentry and I. Mark's going to talk to you about his PhD work. And so with that, Mark, I'll turn it over to you.
All righty. Thank you, Dr. Lewis. Let me get my screen shared here. Get this pulled up. All right, so as she mentioned, I'm going to talk to you about my PhD work. I'm going to throw in some master's level or master's degree data related to the agronomic stuff because I feel that a longer look at yield and things like that is super important. So as she mentioned, I'm going to talk to you today. Title of my presentation is Soil Management Affects Soil Biochemistry and Agronomic Production. So a little bit of introduction to the sort of issues we face out here. Soil erosion has been an issue across the US for decades, especially on the Southern High Plains, the area around Lubbock. Wind erosion really peaked during the Dust Bowl of the 1930s. However, since that time, we have been able to significantly decrease erosion, mostly thanks to conservation farming practices of our producers, and they've really found that crop rotations, crops and no tillage do a really good job of mitigating our wind erosion. In addition to that, cover crops have also been reported to increase soil organic carbon, as well as increase microbial activity and biodiversity, and increase water infiltration and water holding capacity. However, on the high plains, like a lot of areas across the country, producers are often reluctant to implement some of these newer practices or these ideas. And the main few reasons we hear out here are that the cover crop, especially a winter cover crop in our case, will use water over the winter and reduce the available water for their planting in the spring. Secondly, cover crops cost money, right? So you have to do that initial investment into the seed to hope to have some benefit. And without a lot of information on the books in terms of the first few years of implementing cover crops, a lot of producers tend to be a little bit hesitant to make that upfront investment. And lastly, there has been some data that reports potential yield loss with the use of cover crops. And so we wanted to kind of also look at that and see if that was truly the case out here on the high plains. So as I mentioned, cover crops and no tillage can change the soil microbial activity as well as affect the soil chemistry and nutrients available. And one of the ways that they have been shown to affect or be affected by those changes in microbial activity is with gas emissions. So measuring gas emissions with these new conservation system implementations will help inform future agriculture production decisions with agronomic and environmental sustainability. So we know that soil and plant respiration is a source of CO2, which is involved with climate change through the greenhouse effect. In addition to that, denitrification occurs in our soils, in all soils for the most part, and represents a potential loss of nitrogen for plant use, as well as a potential effect on the atmosphere through the production of nitrous oxide. So nitrous oxide is a greenhouse gas with a global warming potential 300 times that of CO2. It's present in much less quantities than carbon dioxide, but with a 300 time warming capacity, it still is a pretty significant thing, and it's mainly produced through agricultural soil management. So we can sort of measure the potential of our soil to produce N2O through looking at the abundance of denitrification genes and the potential for loss of nitrogen as N2O or N2. The two main ways, or the two genes or two sets of genes that we typically look at, are NERK and NERS. which do the reduction of nitrate to nitric oxide, which leads to N2O production within denitrification. The second set of genes we typically look at are NOSC clade 1 and clade 2, which are responsible for the reduction of N2O to N2. This is really the ideal fate of soil nitrate being lost as a gas. We don't want to gas it off as N2O because of its effect on the atmosphere, as well as we can gas off nitric oxide in some cases, which contributes to not only the greenhouse effect, but also will can be involved with acid rain. So the objectives of my study were to quantify the changes in lint yield, fiber quality, and agronomic nitrogen use efficiency for cotton crops with cover crop and altered nitrogen fertilizer application implementation. And then the second objective was to assess changes in soil chemical characteristics, greenhouse gas reduction, and microbial abundance during this transition period. So to tell you a little bit about the study, it was conducted from 2015 to 2019, or at least that's the years that are fully complete. It is still being conducted as of now, but that represented the first 4 years of cover crop implementation. And so the agronomic measurables we looked at for this study were yield, fiber quality measured via HVI, as well as seed nitrogen content and agronomic nitrogen use efficiency. So as I mentioned, it was implemented in November of 2015, and the site had previously been under conventional tillage for at least 60 years. So this really represents a long history of conventional tillage and typical farming practices for the high plains, and then trying to see what happens when we switch to these sort of more conservation systems. The study design was a split plot with 3 reps, and the main plot was tillage systems, which there were 3, and you can see those in the left picture here, where we had our no tillage with a winter wheat cover crop, our no tillage winter fallow in the center, and then our conventional tillage winter fallow on the right. Then our split plot was nitrogen treatments, of which there were 4. We had 100% pre-plant, which was obviously applied prior to planting cotton, 100% side dress application, which was applied at pinhead square, or the first reproductive structure of cotton. Then we had our split application, which was 40% pre-plant and 60% side dress. And lastly, we had our 100% pre-plant application with a nitrogen stabilizer product. And then we also had our no nitrogen control as our baseline. To get into a little bit of methods for this study, soil samples were collected at key cotton growth stages for 2018, 2019, and 2020. I'll be presenting just 2018 and 2019 data, as I'm still finishing up collecting the 2020 results. So the 3 growth stages we chose were vegetative growth, which was in June. Peak plant production on the high plains for cotton crops tends to be near August. And then reproductive growth, boll fill, and finishing out those plants in October. And these soil samples were analyzed for carbon mineralization via 3-day CO2 flux, inorganic nitrogen via KCl extraction, gravimetric water content, and pH. We also took gas measurements monthly, as well as 7 and 14 days following nitrogen fertilizer application. When possible, we did have a few technical issues with our instrument during the first 2 years of the study. So you will only get to see a few seasons for those, but we do have a full data set for 2020 that's currently being analyzed. To round out the methods here, talking about our microbial gene abundance. Gene abundance was analyzed with quantitative PCR for the key bacterial denitrification genes, as well as total bacterial gene abundance, as I mentioned before. Total abundance was measured with 16S, and denitrification abundance was measured with those 2 gene sets I mentioned before, NIR-S and NIR-K, as well as NOSC clade 1 and clade 2. Statistical analysis was conducted in SAS, and principal component analysis was conducted in R. So getting right into the results here, Tillage system affected cotton lint yield in all 4 years of the study so far. So in the first year, we saw that the conventional tillage system produced more cotton lint than the no-till wheat and no-till systems, but that was also a really high yielding year. It was really the last typical year in terms of climate, or the last, I don't want to say good year, but kind of average year compared to the 30-year averages. 2017, 2018, and 2019, we saw reduced rainfall over the winter and in the spring, which could have decreased our emissions or our yield, but overall, our yields were pretty average compared to yields for the area. So starting in 2017 and continuing on through the study, we saw that the no-till wheat system outperformed the conventional till systems in terms of lint yield, with greater increases for the no-till wheat compared to the conventional till system seen as the system matured. In addition, in 2019, the last year we measured, the no-till wheat system also outperformed the no-till system, which may indicate some compounding benefits of the no-till wheat system compared to just a no-till without a cover crop. So secondly, cotton fiber quality. We measured all the cotton fiber quality parameters, including fiber strength, micronair, fiber length, and uniformity. Fiber strength was affected by tillage system in 2017 and 2018, and it's represented here with fiber strength on the y-axis and the years in tillage systems on the x-axis. I didn't mention this before, but for all graphs showing tillage systems, the no-till wheat system will be maroon, the no-till system will be gray, and the conventional till system will be teal. So as I mentioned, fiber strength was affected in 2017, 2018 with different patterns. The no-till wheat system had greater fiber strength in 2017 compared to the conventional till system, and the conventional till system had greater fiber strength than the no-till wheat system in 2018. Micronair was also affected by tillage system in 2017, with the no-till wheat and no-till system having greater micronair than the conventional till system. And micronair is an estimate of, or a proxy for, cotton fiber maturity, which really affects the dyeing process and is one of the key things that spinners and I guess consumers down the line look for in a cotton fiber. In 2017 and 2018, all the values for micronair were in the discount range, which doesn't bode well, but they were pretty commonly low across the high plains in those 2 years. However, in 2019, micronair was greatly improved with an average of 4.26, which was slightly above the average for the area and placed our cotton fiber nicely sort of in the middle average, no discount range for micronair. As I mentioned, we did measure fiber length and uniformity, but neither of those were affected by our implemented tillage systems or nitrogen treatments. Cottonseed nitrogen is a pretty good measure of our cotton uptake. Seed nitrogen typically represents about 60% of the total cotton nitrogen uptake. So it's a pretty good measure of how our plants are doing in terms of using the nitrogen we're giving them. Seed nitrogen was only affected by our nitrogen treatments in the 3rd and 4th year of the study, where we saw reduced concentrations for our control compared to our applied nitrogen treatments. This was pretty interesting because in the first 2 years, we didn't see this reduction, which could be due to residual nitrogen in the soil profile, as well as a couple other factors that we'll talk about. However, in 2019, we also saw that our pre-plant and our side dress treatment, those single application treatments without a stabilizer, were not different than the control. And this could be pointing towards the system kind of evening out, or those two single application treatments not allowing for the greatest possible nitrogen uptake for those cotton plants. So lastly, for our agronomic results, we'll talk about agronomic efficiency. It was calculated within tillage system due to those tillage system effects on yield, and the timing of nitrogen fertilizer application didn't affect agronomic efficiency in the first 4 years of the study. Agronomic efficiency was generally low for our nitrogen treatments fall after 2016. And that really means that our control is doing just fine without us applying nitrogen. This could be due to, as I mentioned, a variety of factors, residual nitrogen deeper in the soil profile, but we also did measure the amount of nitrate in our irrigation water, which added about 50 pounds of nitrogen a year. So that could definitely be affecting not only the total amount of nitrogen applied, but also because we irrigate more frequently than we apply nitrogen, it could be changing up how the cotton plant is actually taking up that nitrogen and when it's taking that up. And then just down at the bottom, you can see the calculation for our agronomic efficiency, which, as I mentioned, it's comparing the nitrogen treatments, added treatments to the control, and then dividing by the rate of fertilizer application. Moving on to some of our other types of results. First, we'll talk about gross CO2 emissions. CO2 was generally increased for the no-till wheat system compared to the conventional till system for nearly all the seasons with data collected so far. So on this graph here, you see CO2 carbon rate on the y-axis, and then our seasons and tillage systems on the x-axis. And then also, as you can see, in 2018, we only got to measure the spring. In 2019, we got to measure the spring and summer. And I'm happy to say that in 2020, we got the spring, summer, and we will have fall emissions data as well. One thing we did notice is that as the system matures, kind of like with yield, we saw that there was a greater difference between the no-till wheat and conventional till system in terms of CO2 emissions. This, again, could be due to the additive benefits of that no-till wheat system, in this case for soil microbial activity. So we're building a community, and it's becoming more diverse and larger in those systems. And so it could be that's just increasing soil respiration in general. One thing to note is that this is gross CO2 emissions, and so it doesn't take into account the carbon fixed by the wheat cover crop over the winter. So our total carbon balance will be calculated for 2020 when we can get an accurate cumulative emission for the growing season. Next, we'll talk about N2O emissions. N2O emissions across the few years of this study generally followed fertilizer application by about 14 days. So we see spikes of N2O in the spring for pre-plant applications. We see that continue a little bit in the summer, but then we start to see some side dress applications in 2020 spiking in terms of N2O emissions, as well as our split application kind of being in the middle for spring and summer. Emissions for N2O are always highly variable, especially in arid systems, because N2O is produced through denitrification, which is an anaerobic process. We related N2O emissions to temperature as well, and they are fairly related to temperature, but not nearly as much as CO2. There was little to no N2O production in our control where we weren't applying nitrogen. And then we also determined N2O consumption at various points across all 3 years during those seasons. But when we average by season, we tend to lose a little bit of that N2O consumption. But we did see it in the summer of 2020 definitively in the control. And that N2O consumption is likely related to non-denitrifying bacteria capable of N2O reduction to N2. And so a little callback. So the N2O reduction to N2, if you remember, is done by clade-2 in denitrification, and specifically, or by NOS-Z and specifically by clade-2 in non-denitrifying bacteria. And we'll talk about that in just a little bit. So moving on to some soil data. First, we have carbon mineralization. General patterns increased for no-till wheat systems compared to conventional till systems in 2018. And then in 2019, there was no real general pattern. Inorganic nitrogen was increased for conventional till systems in both years during at the peak sampling. And this could be due to a variety of factors, including cover crop use of the nitrogen immobilization by the cover crop, and also reduced plant uptake in the conventional till systems. pH saw no patterns really between the 3 different tillage systems. but we did see a general increase in both years throughout the growing season, which is likely due to the alkalinity of our irrigation water. And lastly, gravimetric water content is variable between years in terms of where we see the greatest gravimetric water content, but that's really affected by when our precipitation and irrigation events occurred. However, we didn't really see those changes for the no-till wheat system compared to the conventional till system that are typically seen. So talking a little bit about our gene abundances for those denitrification genes, presented here as relative abundance or %16S, so percentage of the total bacterial population that would also have one of these genes. So we see that for NER-S, NER-K, so those N2O producing genes, or those genes that produce enzymes to produce N2O, as well as the NOS-C clade 1, they're less than 1% in terms of total bacterial abundance. However, with NOS-C clade 2, we see that it's nearly 40% in 2018 and nearly 20% in 2019 in terms of the total bacterial abundance. And this is kind of what I was talking about earlier, where that NOS-C clade 2 is present in a lot of non-denitrifying bacteria. It's maybe even ubiquitous in nature due to its use as in a bet hedging strategy for those microbes. So what happens is in soils, also in wastewater treatment, when the environment goes quickly from aerobic to anaerobic, such as after precipitation or an irrigation event, a lot of those microbes need to then switch on and start doing anaerobic processing and anaerobic living. That's very energy expensive. There is a large contingency of microbes that have thus taken hold of this NOS-C clade 2 gene to produce an enzyme to reduce N2O to N2 during those times. And then when the soil quickly switches back to aerobic conditions, they have spent less energy to survive during that time, still produced a relatively large amount of energy compared to normal respiration, and they can move on with their lives. So relating all of this soil and gene data together in a principal components analysis, we see a slight clustering with our different samplings across the 2 years. But most interestingly, we see this clade 2 NER-K vector coming close with our carbon mineralization and pH. So we know from the literature that carbon mineralization and pH really control denitrification and also control how much N2O is produced. And especially in arid soils where maybe our anaerobic events aren't due to irrigation or precipitation, but due to greater soil respiration with greater soil organic carbon, such as where there's a greater carbon mineralization. And so it's pretty neat that over the 2 years of the study, we were able to really show that that is not only the case in other areas, but also is the case on the high plains. So to discuss quickly some of the results we've seen here, no tillage with a cover crop increased cotton lint yield in the last 3 years of the study. And we didn't really see any negative effects on fiber quality, which is great news for our producers. There's no nitrogen fertilizer timing effects on yield or agronomic nitrogen use efficiency, but that could be due to the addition of nitrogen through irrigation water or residual nitrogen deep in the soil profile that we really weren't able to measure. Our soil chemical characteristics varied with temporal sampling, where we saw inorganic nitrogen affected by our tillage system. We didn't really see any effects due to nitrogen, our nitrogen treatments. pH wasn't really variable within the growth stage of the cotton plant, and we didn't see those expected changes in gravimetric water content. Denitrification genes were relatively unabundant or inabundant compared to total bacterial abundance. However, we did see that NOS-C clade 2 was pretty highly present across the bacterial population. And again, that's due to the bet hedging strategy of those non-denitrifying organisms. The presence of this clade-2 may also result in N2O consumption later in the year, or when soil N is limiting, such as near the end of the growing season. And so really could represent a way for farmers to mitigate their emissions and also potentially capture some nitrous oxide and reduce those concentrations in the environment. As I mentioned before, there's no tillage or treatment effect on clade-2 abundance, but it would allow for the potential of N2O consumption in semi-arid systems. PCA analysis of those denitrification genes and soil characteristics gave us some pretty interesting and I guess affirming results that greater C could lead to more respiration and less oxygen conditions, selecting for those bet hedging microbes, as also a potential for us to control and manage for carbon mineralization, resulting in more N2O reduction. So kind of in conclusion, cotton lint yield was improved on the Southern High Plains and those newly implemented systems. CO2 emissions were affected by tillage system. N2O emissions were affected by N-fertilizer application. And soil chemical characteristics and denitrification gene abundance change within the growing season. Overall, I would say the implementation of no-till wheat systems is beneficial to Texas High Plains cotton production, not only for the agronomic side of things, but also for the potential for greater soil chemical changes, as well as soil microbial activity. Some future directions for this project. There's a second research site located in Chillicothe that was also sampled with the same scheme. Their third year is currently being sampling is currently being conducted. We also measured in-ground soil gas concentrations with a modified soil gas probe that we developed with that publication pending. We measured, or we're going to look at whole genome sequencing to help us better identify some of these N2O consuming microbes. And we'll also be measuring NOC activity with transcriptomic analysis. With that, I would like to say thank you to a lot of people. My advisor, Dr. Katie Lewis, she stuck with me through my master's and my PhD, so I'm very grateful to her for that. Her entire group in Lubbock, they've been such a great help, and it's been a lot of fun over the last few years, so I'm very grateful to them. My co-advisor, Dr. Terry Gentry at A&M and his whole lab group really helped out a lot. Dr. Paul DeLon and his lab group for managing our second research site, as well as being a great mentor to me, and Dr. Thomas Boughton, who has been there to answer so many questions. I would also like to thank Texas A&M AgriLife Research, both for their strategic initiative assistantship and their air quality initiative for funding. And with that, I'll take any questions.
Great job, Mark. Dishing the dirt on tillage and nitrogen. Questions? Step right up. You seem to have a lot of nitrate in your irrigation water.
Yep.
Where do you get your irrigation water from?
The ground. So it's the Ogallala Aquifer. So.
Oh, wow. So the nitrogen is getting all the way down into the groundwater.
Yeah, I think it's a pretty common issue for a lot of aquifers across.
Okay. And where do the people get their water from to drink?
Also the aquifer.
Huh. Nitrates, not so great for people, are they?
Nope.
Don't like that. Questions?
Yeah, just out of curiosity, where do you see it going as we go forward? How do you see it fitting in?
Could you be more specific, Dr. Baldensberg?
Well, what's your vision for the next steps and how it might be applied?
I think for the High Plains specifically, I think continuing to convert to some of these more conservation practices would be ideal in terms of being more sustainable for the environment. And my research is just a small part of all of the research Dr. Lewis is doing in terms of evaluating the sustainability of these systems agronomically. So my study shows that no-till wheat can increase cotton lint yield compared to conventional tillage. But we have another study on a different soil type, slightly south of Lubbock, that says no-till with a cover crop may not increase yield. Some studies show that fiber quality is affected, some studies not. So I think continuing to look at these and just encourage the best practices possible on a farm by farm basis is super important.
Other questions?
I have a question. What is the percentage of the farmers that are using no-till?
So across Texas, I believe I want to say it's using no tillage is much more common. It's less than 20% using no tillage with a cover crop. And I believe no tillage also includes dryland acres that farmers won't go through until. So I think it's more common. I don't know the number off the top of my head. I wouldn't want to speculate without.
Just a follow-up question then. So with your results, you would suggest them to apply no-till, right? So how would you assess them to say, okay, you should not till your soil? So how would you approach that? How would you convince farmers that no-till is actually the way to go?
I think the data really speaks to that with those increases. One thing I didn't mention is one of the main reasons we think we see those increases in yield is due to early season protection. So with cotton seedlings out west, our springs tend to be really hot and dry the last few years. And so those cotton seedlings sitting on top of a seed bed are pretty exposed and they can be hurt by wind erosion pretty severely. So when you have that wheat, you're kind of building that wheat residue there, you're kind of building a little house for those cotton seedlings to establish. And that really, we think that's one of the main reasons why we're seeing yield increases. And the main things farmers are concerned about, right, are yield and then also the quality of their cotton fiber. And we show that you don't really see a reduction in cotton fiber quality. So you're not going to get it dinged on sort of the end of your production, but you're also going to hopefully see greater or equal yield.
All right. Thank you, Mark. Great presentation. Thank you very much.
Thank you.
Other questions?
Mark, this is Reagan Noland out at San Angelo again. I was curious about the differences that you saw in fiber quality. Like you showed some responses in strength to different cropping systems or tillage regimes. What physiologically in the plant, what do you think drove those differences? Was it nitrogen or is there something else? Or I'm learning a lot about cotton currently, so I'm just curious.
I am definitely not a cotton physiologist, but from what I know, a lot of fiber quality parameters are obviously number one genetic, so they've improved over the years substantially with the greatness of our plant breeders, but also late season moisture has a lot of effects on that boll developing and then drying out. And so with micronair, especially late season moisture can reduce the maturity of the cotton fiber. So that's one thing that producers and people have been concerned about with the no-till wheat. So if you've got a lot of wheat residue and you're keeping that soil moist when you get those late potential for late season rains, then you can reduce your micronair. But we didn't really see that, which was pretty good. And I think that just speaks to, I guess, the arid nature of our environment.
Yeah, I would agree with you on that. I'm more concerned with water conservation than with quality when it comes to having water versus not in the soil. So I like having the residue out there too. But that's really interesting. I actually I didn't know that about that relationship with Mike and kind of seasonal moisture availability. But anyway, really, really cool stuff. Thank you.
Appreciate it.
Well, that's great. I'd like to thank our presenters again for doing such a great job. And we will see you all back here again, same time, same channel next week on the Soil and Crop Sciences Hour of Power.